The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone. My name is Dr. Kimberly Arnold, and I'm the Senior Marketing Specialist for Molecular Biology Services at GenScript. It's my great pleasure to introduce today Marcelo Caraballo, a Senior Scientist from Custom Array, to present today's webinar entitled Oligo Pools, Design, Synthesis, and Research Applications. During this webinar, Mr. Caraballo will provide an overview of oligo pool synthesis technology and illustrate the key features and advantages of using an oligo pool for various research applications. Marcelo Caraballo is a senior scientist at Custom Array located in Seattle, Washington. For the past 18 years, he has worked to research, develop, and commercialize novel oligonucleotide synthesis chemistry. By combining semiconductor technology, electrochemical techniques, and a traditional oligosynthesis chemistry, he has helped to realize a novel in situ array oligonucleotide synthesis platform. Along the way, he has developed a breadth of experience in both the technical aspects of oligosynthesis as well as their various applications. To ensure that this webinar has high sound quality, we ask that all attendees put their microphones on listening-only mode. Also, please feel free to type in your questions in the chat box in the lower right corner of your screen throughout the talk. Mr. Caraballo will address your questions at the conclusion of the presentation. Now, without further ado, let's welcome Marcelo Caraballo to present today's webinar, Oligo Pools, Design, Synthesis, and Research Applications. On to you, Marcelo.
Thank you, Kim. And thank you to everyone for attending this presentation on GenScript's Oligo Pools service. I'm a senior scientist at Custom Array. Custom Array has developed this technology and recently was acquired by GenScript to bring this technology into their portfolio. And I'm happy to share it with you today. So today we're going to be talking about oligosynthesis technology, the design of oligos for applications, and those applications themselves. Now, as a caveat, I personally am largely interested in the synthesis technology. That's what my expertise is in. That being said, I have been exposed to a lot of these applications, and I'll be happy to share anything that I picked up along the way. If there are any questions at the end that I can't answer specifically about these applications, I'm sure we can find someone at GenScript who can. Just feel free to contact me or Kim, and we'll get back to you as soon as possible. So, as I said, we're going to start off with the oligosynthesis technology itself. So, what exactly is an oligo pool? That's the main question. That's what this presentation is about. Well, using an in situ array synthesis technology, we can synthesize many oligos simultaneously. Basically, we're trying to do the same kind of parallelization that is used in NGS, that's revolutionized that particular field. We can do something very similar with oligosynthesis. Instead of synthesizing one oligo at a time, we can synthesize many oligos at a time. And at the end of this entire synthesis process, what you get is a library of thousands or hundreds of thousands, or really there's no limit, it's just how much you want to put into it, of oligos that are completely defined by the customer. And this is all at a tiny, tiny fraction of the cost you would have to spend to make each oligo individually using the traditional oligosynthesis techniques. Now, array synthesis. Well, there's basically three main branches of array synthesis. There's the electrochemical, and that's what we do, and we'll be talking about that in depth. But there's also a couple other ones. There's light-based chemistries and inkjet printing, and we'll touch on those a little bit. But all of these array-based syntheses have to answer one central question. How do you spatially segregate chemical reactions on a planar or mostly planar surface without using physical containment like walls? And the reason why we want to avoid that kind of walls or that kind of physical structure is that it adds a lot of cost. It adds a lot of space. We want to be able to do as many chemical reactions in as small a space as possible. And when you start doing physical micro fluidics, you start having a lot of problems because you can only get so small using those kinds of methods. Now, very quickly, the light-based chemistries. This is kind of the granddaddy of array synthesis. One of the first ones was developed. This particular method used custom phosphoramidites. Phosphoramidites are the individual building blocks, the A, the C, the G, the T, along with some light-sensitive protecting groups that allowed the use of localized light. The first one was photolithography. These are basically opaque masks with small holes in them, DLP or digital laser projection, or some other kind of laser light to be able to localize light in one spot to allow the chemistry to occur in that spot and not in other spots. This was a great early entrance of array technology back in the late 1990s. It unfortunately suffered from being very expensive because of the custom phosphoramidites and also very poor synthesis fidelity. It was first, but it has actually kind of moved on from there. We don't really see this this method being very used anymore. There's also inkjet printing. That's another method. Basically, inkjet printing is exactly what it sounds like. You use a very sophisticated inkjet printer, but instead of printing your standard inks to print a document, rather you print reagents, and by controlling where these reagents are placed on a planar surface, you can localize the chemistry to just those areas. This has some pretty good advantages in that you can use off-the-shelf reagents, but it also has some disadvantages because anyone who's used an inkjet printer knows that they can be fiddly things. They require some maintenance, they require very specialized nozzles, and they're a workflow bottleneck. Anyone who's worked in an office where everyone wants to print something, and someone can very much hog up that machine for quite a long time. So it tends to have a very slow synthesis time. Moving on, electrochemical is what we do, and it has its own advantages that we bring to bear. Now, unlike the inkjet printing where the actual technology is the machine itself, for us, the technology is not really the machine in and of itself, it's the chip. So we have a very sophisticated chip in a very simplistic machine or synthesizer. The advantage there is that it's very modular. We can add machines or remove machines, and the chip itself, it's advanced, but we're also leveraging the semiconductor industry, which does tens of thousands of chips or millions of chips every day. So we are able to leverage their kind of reproducibility, and it helps us quite a bit. We also have the fastest turnaround time in the industry, and that goes back to the kind of the modular workflow that lets us be very, very quick. As soon as we get an order, we're able to kind of put it on a machine and get it going very quickly instead of having to wait and batch into larger order sizes. So electrochemical oligosynthesis using CMOS technology. For those who don't know, CMOS is basically stands for complementary metal oxide semiconductor technology. That's a lot of words. It's not really important, but basically what it means, this is the same kind of technology that builds all of the world's computer chips. We're basically building DNA on computer chips. The way these things are made is that you start with a silicon wafer. This is made by what's called a wafer foundry. And the wafer foundries that we use are the same ones that are used to make computer chips that go into computers or your iPhone or whatever. We have these wafers made in large batches, and then these individual rectangles you see here, these are what are called die, which we eventually call chips. These are the individual chips that get placed in a package. This package basically is just designed to look like a microscope slide, and that's just because there's a lot of automated tools that let you manipulate microscope slides easily. It could look like anything, but this is what we chose to make it look like. And here you have a blow-up of what the surface of the chip looks like. This surface, what you see here, is a 3x3 grid of platinum electrodes. These electrodes are where the actual synthesis occur. By turning on and off the electrodes, we can manipulate which or what sequence is made on which electrode. Over here you have the actual synthesizer itself. As I said, it's a rather simplistic machine. All it really is a fluid mover. The fluid gets moved into the reaction vessels, reaction chambers, and the chip does all of the actual advanced work. But before we go on get too far down that line, I just wanted to have a short primer on oligosynthesis in case this is not something that is familiar to other people. The current incarnation that we use in oligosynthesis dates back to the Carruthers lab at the University of Colorado Boulder in the 1980s in his landmark paper. There's been a few changes here and there and some advancements, but it's a direct lineage to that foundational work. In that work, he basically sets out this molecule. This is a phosphoramidite. It's or a monomer. This is an A phosphoramidite. So there are certain common features. What you see here is a number of protecting groups. The protecting group in the big red circle, that's called DMT, that's dimethoxytrittyl. That is the one that protects the 5' and keeps the chemistry from going forward, so you can selectively add one base after another. And there are some other protecting groups that are placed in here to keep certain reactions happening throughout the chemistry. There are 4 basic steps. This is true basically of all phosphoramidite synthesis, all oligosynthesis in the world. You have detritylation, activation or coupling, capping, and oxidation. And you kind of repeat this over and over again to make a full oligo. All oligosynthesis, or most oligosynthesis in the world, is going to be 3' to 5', which is not as familiar to molecular biologists, where it's usually the other way. But in chemical oligosynthesis, it's 3' to 5'. There are some reverse chemistries that allow you to do it the other way, but they're relatively bespoke and not very common. This next slide shows you essentially the chemical diagram of what is occurring. The first step, you have the first base, and you see that large DMT group. That's the protecting group. Now, that protecting group, the crux, or the important part of this protecting group, is that it's an acid labile protecting group. An acid, you expose an acid to this protecting group, and it is removed from the base, leaving a free hydroxyl. That free hydroxyl is now free to couple to the next base. So now you expose that deblocked base to a new base in combination with something called an activator. There are many activators. This one says tetrazole. That's one of the older school ones, but there are many other ones at this point. And during this process of coupling, the new base is added to the first base. You see there, that's a trivalent phosphite. That's not a stable arrangement. So the next couple chemical steps basically are just going to fine tune that, and you end up with a pentavalent phosphate. And now this looks more like what you want to see. You repeat these steps over and over again to make the oligo. It's a very cyclical process. We basically talk always in cycles when we're talking about DNA synthesis. Now, moving on to how we do it specifically. Well, again, as I mentioned, the trittyl group, or the DMT, that protecting group, is acid labile. So what we do, instead of the normal way, is use a bulk acid like trichloroacetic acid, but that would remove all the protecting groups. We want to selectively remove these protecting groups, so we electrochemically generate an acid. So by turning on this electrode, we can generate the acidic environment in only that area, removing the protecting group of only the oligo being made on that electrode, leaving that free hydroxyl. So that particular base is now ready to have another base coupled to it. And that's basically the crux of the whole technology that we use here. Here you have essentially an image where you have the surface of the chip, and we use, this is for illustrative purposes, this is aqueous and with bromophenol blue added just to visualize the acidic environment. One of the important things you need to do when you create an array technology, as I mentioned earlier, you have to segregate and separate these chemical reactions. So here you have on the left, you have where we have fine-tuned the chemistry to create what we call a virtual flask. The yellow there is where the acid is being generated, and you can see that it doesn't spread to any of the neighboring electrodes. You want to be able to only create the acidic environment where you want to and make sure it doesn't go anywhere else. On the right, you can see what we've purposely made it not work, and here you see this diffuse acid across the entire surface, which is not what you want. As a kind of a sum up of this entire thing, the electrochemical synthesis process, you start with, here you have 2 electrodes. Both of them have a T on them, and both of those T's have protecting groups. The first thing we do with that nice lightning bolt graphic, we turn on the electrode of that first electrode. You turn on the voltage, and that creates an acidic environment just above that particular electrode and not the neighboring electrode. Then, in the next step, we introduce or flood the entire chamber with an A phosphoramidite, again, along with an activator. This is implied. And the A couples only to the T, the first T, and not the second T. Then we wash that away, get rid of all of that, and then we turn on the second electrode that wasn't turned on the first time. There, the protecting group is removed. You see the little red balls are gone. And then in the next step, we add the C phosphoramidite, which again only is coupled to the T, the second T, because the first one was not turned on. So now in the first electrode, you have TA, and in the second one, you have TC. This is basically what we do, only on a much larger scale. Here you see 2 electrodes, but on our chips, we have tens of thousands of electrodes, all of which are being turned on at various points in time to add these bases so we can make many, many thousands of oligos simultaneously. At the end of the process, you essentially have an array full of oligos. The one on the left is a microarray scan. Basically, we took the chip, we synthesized some oligos on the surface of the chip, and then we did a hybridization with some fluorophore in order to kind of visualize some of the oligos on the surface of the chip. This is our 12K chip on the left, and you have a blow up of what the surface of that 12K chip looks like. The specific size of the electrodes of the 12K chip is 44 microns in diameter. And on the right side of the screen, you see the 90K chip. The 90K chip obviously has many more electrodes. We call these 12K and 90K. That's not the exact number of oligos that are available on each chip, but the actual number would not roll off the tongue very well. And then you also see a blow up of the 90K chip on the rightmost. Those electrodes are smaller. They're 25 microns, but there are many, many more electrodes. So how are oligo pools constructed? Well, basically we use that technology, we use the electrode array that I mentioned as a starting point, and we synthesize a unique oligo on each of those electrodes. After the synthesis is complete, you have all of these oligos on the surface of the array, but for a pool, we don't want them there. We want to remove them, so we cleave them off the surface of the array. We do some minor processing, desalting, and some minor purification. And the end product is a tube of oligos. These are single-stranded oligos in TE buffer. We generally ship in 80 microliters of TE buffer. At that point, it's ready to be used as soon as you get it. People often ask about the quality control of oligo pools. Oligo pool quality control is a little bit more complicated than quality control of a single oligo, because you have many thousands of species that are all mixed together. Fortunately, we have a big advantage here compared to some of our competitors in that the synthesis surface itself is a computer chip. It has many different kinds of feedback, of electronic and digital feedback that we monitor. The performance of each electrode is monitored continuously by a number of different avenues, and all these are logged in the synthesis logs, so we can have really high confidence that the synthesis occurred properly. But we also like to have some visual verifications, some other verifications. After the synthesis is complete, we take that chip and we do a fluorescence scan. DNA with the protecting group still on it will fluoresce in certain wavelengths, so we can visually record the synthesis on each of the individual electrodes. Here you see a picture of a small segment of a chip, and there's a black line in the middle. That black line is where we have intentionally left the oligos or intentionally left the electrodes blank with no oligos synthesized on them. It provides a nice contrast line that's used as part of our kind of a quick QC check. After that, like I said, we did some purification, and then we run it on a nanodrop to get some nice measurements of the ratios and the yield of the pool itself. Generally, the next step after that is a QC-RPC. This is done from time to time, and it's also something that our customers do very often. Here, you see what a PCR looks like of an oligo pool. What we've done here is that we've constructed an oligo pool that has sets of different lengths of oligos, 80, 90, 100, 110, 120, and 130, and we synthesized it on a 12K array. Now, all of these different lengths of oligos all have the same universal flanking priming sequences, so we used a single pair of primers to amplify all of these subsets. And at the end, you see a nice step ladder effect of the different lengths of oligos, the different groups of the oligos that we built. This is something that's very, very, very common for us to do, and it shows like a nice bands that you want to see of the correct length. Some quick data on our performance. This is a stability study. It wasn't performed by us, it was performed by a customer. They were interested in this question, so they asked us to take a file that they designed and synthesize it over the course of 8 weeks. And the goal was to use different synthesizers, different reagent lots, different operators, different chip lots, about as many different variables you possibly could get, just to see how stable everything was. And the results were very good. At the end of the process, we sent them all 8, and then they took it and did sequencing, and they were kind enough to give us this chart of data and allow us to share it with everyone. This basically just shows you the error rates, the distributions of the different oligos, and the recovery rate. If anyone has any questions on this afterwards, we can go back to it. This is a distribution case study. Basically, this is somewhat similar to the previous slide, but this is one we conducted internally. Customers sent us an order of 12,000 unique oligos. These were 125 mers. We made it on our 12K chip. And then we took a kind of a deep dive into it to look at what the performance of these oligos were. And you'll see that the distribution is very tight. It kind of centers around a very, very tight bell distribution there. And the error rate of this particular oligo pool was very, very good. It was 0.27%, or 1 error in 372 bases. Error rates kind of fluctuate a little bit depending on the individual designs, but this was a pretty good example. So moving on, we're going to talk a little bit about the design aspects, some minor design aspects for the various applications. As I mentioned earlier, I'm no expert in these things, but there are some kind of universal things to talk about. Almost all oligo pools are intended to be amplified, and there's a couple of pretty good reasons for this. The main reason, the one we'll talk about a bunch, is purification. So it's all oligosynthesis, whether that be a primer or that be all our oligo pools or anyone else's oligo pools, suffers from stepwise truncations. What happens is that none of these chemical steps, remember this is a cyclical process we're doing over and over again, these chemical steps do not go to 100.0% completion. So you start with a certain amount of starting sites or starting scale, and then as you add base, every time you add a base, you lose a small percentage of your starting material. So at the end of the process, you have an electrode with 3 full-length copies, but then you have a series of truncated copies. This is obviously an exaggeration, but you have some full-length copies, and then you have some shorter and shorter truncated copies. These HPLC traces you see here compare and contrast standard oligosynthesizers that would make a primer and our synthesizer, both with just a poly DT as well as with a heterogeneous 35 mer. And you'll see when you start with just a DT, well, that's really easy. That's a single base-base combination that can have a very high coupling efficiency. That typically we call this a coupling efficiency. But when you start mixing different bases, these different bases have their own unique effects. So the coupling efficiency generally goes down with when you talk about a heterogeneous mix of oligos and bases. So as you get longer and longer oligos, your percent full length will go down. In this chart here, a 60-mer will have roughly 50% full length, and the other 50% will be kind of a tailing off of shorter and shorter truncated products, assuming a 99% stepwise or coupling efficiency. So the presence of those shorter or truncated copies can be very messy for a lot of applications. So by purifying it, you kind of go, by amplifying it, you purify it, and we'll go into a little bit about that, how that works in the next slide. But a couple other reasons why people generally will amplify an oligo pool is that there's relatively low copy numbers. I mean, these electrodes are teeny tiny. They're 44 microns or 25 microns. The starting scale of the 12K electrode is only 1 femtomole. So you're not working with a lot of copies there. So PCR does a really good job of amplifying this. It makes your pool stretch much longer compared to just using the pool without amplification. And then the last reason is one we touched on earlier, is that it's a great QC. And I'll go into the next slide, kind of covers that a little bit more. So, the first section here, you have an example of a full-length oligo and not a full-length oligo. Remember, DNA synthesis is 3' to 5', and that's why it's written in this particular way. All of the copies will have the 3', but not all of the copies will have the 5'. The ones that don't have the 5' just won't amplify it. The 4' sits down, it's going to make the second strand for both of these, but then the reverse primer won't pick up the truncated copies. It'll only amplify the full-length copies. So the PCR basically will be exponential for the full-length copies, and there'll be some small linear amplification of the truncated copies, but those fall by the wayside very, very quickly. So PCR is a great way of purifying an oligo pool. It also has another benefit. The other benefit is that it allows you to arbitrarily subdivide a master pool into a number of subpools. These are basically methods, we usually call these cohort primers. Instead of having a single priming sequences for all the oligos in a single pool, you can subdivide that and say, this 1,000 will have this priming sequence, and these 1,000 will have these priming sequences, and then use different PCR primer pairs to select and amplify one subpool out of the master pool. Many times, customers don't need 12,000. They only need 6,000 for one experiment and 6,000 for another experiment. That allows them to order only one oligo pool, and then you use one primer pair for one experiment and a different primer pair for a different experiment. It increases the cost efficiency in a nice way. So finally, we're going to move on and talk about applications of oligo pools, and there's a number of them. So what are oligo pools used for? The first one, genome editing libraries. This is kind of a big one. We do a lot of these now. CRISPR screening libraries are very, very common. Related shRNA screening libraries are also pretty common. They are used for different purposes at different times, but both of them have utility, and both of them use oligo pools. Targeted sequencing. Targeted sequencing is obviously something that I'm sure everyone's familiar with, but they pretty much all use oligo pools. There are different styles of targeted sequencing. Hybrid capture is one of the older and most common ones. There's also MIP probes or MIP style targeted sequencing. That's also another one. There's a few other ones as well. Mutagenesis libraries, again, something that I'm sure a lot of people are familiar with. Oligo pools have a lot of advantages in being able to give you specific sequences there. Oligo fish or merfish, there's a few different styles of fish type applications or in situ hybridization applications. One that's a little bit sci-fi but a lot of fun is DNA data storage. This is the concept of using DNA as a data storage medium. We'll talk about this one in a later slide. And then finally, the one on this list is MPRA, or massively parallel reporter assay. This is kind of a catch-all term. Whenever you have the ability to encode a huge number of oligos and then use NGS to decode that data, you can design a lot of really cool and interesting custom assays that will give you a lot of information. MPRA is kind of a catch-all term. I think there's a lot of different flavors of this one. Going back to the CRISPR application, again, this is not one that I'm an expert in. I'm sure many of you have much more expertise in CRISPR in and of itself, but we've been involved in the CRISPR community for quite a while. These are two landmark papers put out by Eric Lander and Feng Zhang at the Broad Institute. These were done using our technology, and basically they were doing human knockout lines. They targeted the entire human genome using a CRISPR library they created using our technology. In fact, many, I should say, CRISPR libraries that are commercially available from Addgene, from other places, were actually made off of custom array oligos that we originally made for them. Gene variant libraries, I think I have another slide. Yeah, I'm going to put them both up there. Gene variant libraries, this is one I'm, again, not an expert in, but when we can synthesize thousands of variants at a time, there's a lot of powerful things you can do that. Instead of doing random degeneracies, we can specifically decide where you want your mutations, how you want them to be, what relationship you want them to have to each other, or proportion. This is really great for protein engineering in a more targeted way. Industrial enzyme development, similar concept. Metabolic engineering. This allows less of a shotgun approach and more of a targeted approach and that oligo pools are great for. There's a lot of different types of gene variant libraries, and we can address many of these different needs. Touching on target enrichment, this is kind of an example of a bait and capture. As I mentioned earlier, there are other methods or other styles of targeted sequencing. Bait capture is one of the older ones. But basically, you start with a pool of oligos that have a universal priming sequence on there. You amplify those, and in the amplification process, you add an RNA transcription sequence, T7 or SP6 or something like that. And then you do an in vitro transcription just to get single-stranded RNA. In the process, you dope it with biotin, and now you have biotin-related single RNA that is complementary to the region of interest that you want to capture. Combining with some magnetic beads in a little assay, you can hybridize these, pull down what you want to sequence, wash away what you don't want to sequence, and then release that from the capture probes. And now you have an enriched sample for the regions you want to do. It's very common. This is something that's been around for a while, and it uses oligo pools. Now, one of the advantages of using an oligo pool directly for doing this is that much cheaper. You can order a single oligo pool, and that is between the process of PCR and IVT. You can have thousands or tens of thousands of enrichment reactions from just a single oligo pool. But it requires a little bit of know-how on how to process an oligo pool from single-stranded DNA to biotin-related RNA that is good for your enrichment process. But a lot of people do this, and it's pretty popular. Finally, DNA data storage. As I mentioned, this is kind of a fun one. So the idea here, the reason why this has become a little bit of an interesting topic is that the leading manufacturers of digital media have basically said we're going to run out of our ability to produce flash drives, hard drives, what have you, DVDs, that based on how quickly demand for storage is going up. This graph here shows you unmet need versus time. And extrapolating out to 2025, we're going to have a huge gap between the amount of storage media that they can pump out and the amount that we want to save. The idea of using DNA as a data storage medium is not new. Obviously, nature has been doing this for a very long time, and it has some interesting advantages. DNA itself is relatively stable. Anyone knows DVD won't last that long. Flash drives don't last that long. Same thing with other magnetic devices. DNA has been sequenced from tens of thousands of years old, or even longer in some cases. So it's a relatively stable storage method of storing data. It's also very compact. Here you have a kind of a fun graphic of a basketball, and basically someone did the calculation, and they said that the entire world's 2013 data output could have been saved in an amount of DNA that would take up that amount of space. Doing the same thing using current digital data mediums obviously required quite a lot more space. So while it is kind of a little bit sci-fi, it is being used. Here are two examples. Nick Goldman at the Wellcome Sanger Trust Institute had a fun thing that he did at Davos in 2015, where he encoded, using oligos we provided to him, he encoded the data for a Bitcoin. And it was encrypted with his encryption technique or algorithm, and he handed out tubes of DNA that we made for him to the audience members at the meeting, and then challenged anyone to decrypt it and solve it to claim the Bitcoin, which it was actually. It was claimed by a postdoc, I believe, who decrypted it and was able to claim the Bitcoin. Of course, the value of Bitcoin is very variable and not too good at the moment, but still, it was a very fun project. In addition to that, there's another example, a company called Turbo Beads. This is work done by Dr. Robert Grass in Zurich, and we made for him a set of oligos and he took that out. He encoded an album, an entire audio album by the band Massive Attack. The album was Mezzanine. He encoded it in using his data algorithm in DNA. We synthesized the DNA for him, and this is something that they've done to show that it's possible. And it's a little bit of a marketing stunt as well. They put out a spray bottle that just had the DNA that we had made for them, amplified PCR product of it, of course. They put a little spray bottle of it that you could order as a collector's edition of the album. Now, this might be a little bit frivolous, but there is a strong argument to perhaps that DNA could be used as an archival storage medium. There is all kinds of things that need to be archived for a very long time that are not generally ever looked at. The read and write time of DNA is not very good compared to flash drives. But from an archival perspective, something that you need to keep for a very long time, where there's a lot of data that you keep for a very long time, it could have a utility there. And it's a lot of fun to work on. So summing it all up, our oligo pool service will give you, maximum screening efficiency. Most of the oligos that you want, almost all of them really, in many cases, it's actually all the oligos that are in the 12 or 90 or a million oligos that are going to show up. Our platform can give you everything you want. The variability is very low, batch to batch. That's been done through work from our customers, and we've been verified by a number of different customers that we keep our batch variations extremely low. It's a very flexible platform. We have 2 chip sizes, but you can use combinations of these chip sizes to do multiple or larger pools. I've made pools that are, used 10 or 20 different 90K for 1 to 2 million oligos at a time. That's not a problem at all. And at the same time, you can also subdivide a smaller chip, like a 12K chip, and I've had people do 100 different sub pools inside a single 12K. We don't have any kind of sequence restrictions or minimum order. You just send us the list of what you want. We'll take it, populate it on our chip, and make it. And we have the best turnaround time in the industry. This is something that we value quite strongly. No one wants to wait for their product. We take your sequences, we put it on our chip, we put it on a synthesizer, and generally, as fast as 5 business days, we often hit that. Most people, I tell them, I'll have it for you next week. That's generally what I try to do for everyone. And for most people, it's a pretty great turnaround time, and it's certainly the quickest we have available in the industry. Anyway, I wanted to thank everyone for attending this presentation on oligo pools. It's something I've been working on for a very long time, and I enjoy my work quite a bit. GenScript has a number of other product lines, and they have a lot of things to offer. At this point, I want to conclude this presentation and turn this back over to Kim.